Hello everyone, welcome to this podcast session where we'll be diving into the fascinating world of biochemistry. I am your host Samuel Brown, and with me here are my friends and final year biochemists. Please guys, can you introduce yourself?
My name is Samson Samuel Sunday.
My name is Pudo Utibe David.
And I'm Faith in
Yeah, so today we are going to be discussing about the importance of Krebs cycle to the metabolism of carbohydrates, protein, and nucleic acid. But before we dive into this discussion, let's have a brief summary about Krebs cycle, shall we? Yeah. So Krebs cycle is a series of enzymatic biochemical reactions leading to the production of reducing agents such as NADH and FADH, and also leading to the generation of ATP, which is produced in the electron transport chain through these reducing agents. This Krebs cycle begins with the combination of azolyl acetate and acetyl-CoA, leading to the production of different intermediates, which are now used in the metabolism of the nucleic acid, carbohydrates, and protein, which we are actually going to be discussing about today. So, Delight, please, can you take us into the importance of Krebs cycle to nucleic acid, please?
We all know that nucleotides are the building blocks of nucleic acids. So, one of the important roles in which the Krebs cycle helps the nucleic acid metabolism is by supplying carbon and nitrogen molecules, which are used in synthesizing nucleotides, such as our purine and pyrimidines.
Yeah, that's right.
Yeah.
And then we all know too that nucleotides are building blocks of nucleic acids, which are our DNAs and RNAs. So from the Krebs cycle, we generate, for a complete Krebs cycle, we generate 6 molecules of NADHs, 2 molecules of FADHs, and 2 molecules of ATP. So for the synthesis of our DNA and RNA, we need a lot of molecules of ATP. So the one generated from the Krebs cycle is not enough. So these NADHs and FADHs from the Krebs cycle now goes into the electron transport chain to produce the amount of ATP needed for the proper synthesis of our DNA and RNA molecules.
Okay, great job. So, Samuel, can you please, dive into some importance of the Krebs cycle to nucleic acid?
Okay, like as my fellow biochemist here has already mentioned a few points, I know that there are many more importance of Krebs cycle. I'm going to dwell in one paramount one. That we all know the Krebs cycle contains series of biochemical reactions and that are being produced. So the importance of Krebs cycle to nucleic acid metabolism is that it helps in the production of carbon skeleton. So this, because they are key intermediates in the Krebs cycle, and these intermediates, as we mentioned, is alpha ketoglutarate and oxaloacetate. So these intermediates now they aid in the production of amino acid. Like alpha ketoglutarate to produce glutamate, which is an amino acid. Then oxaloacetate will produce aspartate, which is an amino acid. And these amino acids will now help in this production of the carbon skeleton, which are like the foundation for nucleoside-based synthesis.
Exactly.
Okay.
So you're saying, in which manner, like how does this oxalic acetate now help in the production of this carbon skeleton? What are the process?
Okay, so this, first of all, like the carbon skeleton, they are cores for producing organic molecule. Like because the carbons will link together. Let me bring an example. The structure of acetyl-CoA. Acetyl-CoA now has 2 carbon skeleton in it because it follows the acetyl group CH3CO. So there are 2 carbon skeleton in it. So the process of it producing the carbon skeleton is like, let's take oxaloacetate. Oxaloacetate on its own would directly provide carbon and nitrogen. It will supply carbon and nitrogen, as the brother here had mentioned, carbon and nitrogen, and this carbon and nitrogen will now aid in the production of this structure, because in alpha ketoglutarate, I said that it produces this amino acid, which is glutamate. And this glutamate now in glutamate and aspartate in purine synthesis, which is a nucleotide-based purine synthesis. This amino acid now will help to bring in guanine and adenine, which will help in the purine synthesis. Now, in pyrimidine synthesis, Now it will bring out cytosine and thymine, which will help in DNA, which is in the DNA, and it will also bring out uracil, which is in the RNA. So this is one of the processes that is.
Wow, great, great, great. Everything I've heard right now just made me more curious about the importance of Krebs cycle to carbohydrate metabolism. Having heard about the importance of Krebs cycle to nucleic metabolism, I'm even more curious about the importance of Krebs cycle to carbohydrate. And I have here with me my colleagues, which are actually going to dive into the discussion, the importance of Krebs cycle to carbohydrate. Please, your names.
Oh, my name is Deborah Gerson.
My name is Prince Will.
My name is Beto Obasi.
Okay, great. Deborah, please, can you dive into the importance of Krebs cycle to carbohydrate metabolism?
Okay, apart from TCA cycle producing ATP for nucleic acid metabolism, TCA cycle also has an important role to play in carbohydrate metabolism through complete oxidation of carbohydrate to produce energy. You know that energy for cellular respiration. Now, here, glucose is broken down by series of 10 enzymatic reaction steps. That's where the glycolytic pathway plays in, where glucose is broken down to produce pyruvate. Now, from pyruvate is oxidatively decarboxylated to produce acetyl-CoA, and acetyl-CoA is oxidized. The oxidation of acetyl-CoA produces energy-rich molecules such as NADH, FADH2, that's the flavin adenine dinucleotide, and these energy-rich molecules enters into the electron transport chain. When it enters into the electron transport chain, they are oxidatively phosphorylated to produce energy. Now you can see that in a phase, when pyruvate comes in, in a cycle, pyruvate comes in, one molecule of pyruvate actually produces 3 NADH, 1 FADH, and 1 ATP. Now, when that is done, it enters into the electron transport chain and they are oxidatively phosphorylated to produce ATP. Now, you can see that one molecule of glucose actually produces about 36 to 38 ATP, which is a good number for cellular respiration.
Okay, great. Now, from what you're saying ATP is also produced, apart from this energy carriers, these are NADH and FADH, ATP is also produced by the Krebs cycle.
Yes.
Yeah, wow.
ATP is also produced.
Wow, that's very nice. That's very nice. Okay, please, Prince, can you buttress more on that point?
From what she said so far, NADH and FADH2 actually have more potential use in carbohydrate metabolism for ATP creation in the electron transport chain. Actually, how this works is that NADH and FADH2 that is being generated in the TCA cycle transfer their electrons into the electron transport chain in the inner mitochondrial membrane. And when this happens, the electron in the inner mitochondrial membrane helps to pump proton from the mitochondrial matrix into the intermembrane space. And when this happens, it creates a proton gradient. When it creates a proton gradient, the proton gradient now helps to power the ATP synthase. And now the ATP synthase now helps to convert the ATP into ATP. And when this happens, enough energy is created for other purposes in the body.
Okay, fine. Now, I remember Faith talking about nucleic acid, and she said something about the NADH providing energy, and we have Deborah saying something about the NADH providing energy. I actually like the fact that Prince, you have been able to portray that point, that you have been able to portray the way at which the NADH actually produce this energy. That was what Faith did not do, and that was what Deborah did not do. So it's a very nice point. So this ATP that is produced, how does it help in carbohydrate metabolism? Because I've heard that ATP is produced through the electron transport chain. How exactly does this ATP help now in carbohydrate metabolism?
Actually, one of the ways it helps is that it helps in anabolism of glucose through a process known as gluconeogenesis. And one of the key roles ATP plays in gluconeogenesis is in a role known as pyruvate carboxylation. And how ATP is used is that ATP is used to convert pyruvate to cellulose catalyzed by enzyme known as pyruvate carboxylase.
Okay, great. So ATP, what I'm saying right now is the ATP produced by the NADH in the electron transport chain can now be used in the pathway called gluconeogenesis. in order to convert pyruvate to oxaloacetate through the enzyme pyruvate carboxylase. Okay, great. So with all this thing I'm getting, I'm actually picturing the fact that the Krebs cycle is a hub. Is a hub that leads to different metabolic pathways.
Murder.
At least to different metabolic pathways, right? Better.
Yes, that's true. Another important of the Krebs cycle to carbohydrate metabolism is the anapleretic and catapleretic reactions. Intermediate of the Krebs cycle are taken out and used as building blocks for the production of molecules like glucose, fatty acid, and amino acid. You can see that citrates act as a substrate for the production of acetyl-CoA, which is then activated for the production of fatty acids. That is for the catapleretic side. For the anapleretic side, the intermediate which were removed earlier are then renewed and helps for the for the Krebs cycle to continue its production. And there must be a balance between this catapleretic and anapleretic reaction for the body for the production of energy, which is needed for cellular, which is needed for for the cell as a whole, and for which the main purpose of the Krebs cycle is an integration into other metabolic pathways.
Anapleretic and catapleretic.
Yes.
Wow, I've heard a lot about carbohydrate metabolism, and I know that there is more to talk about in this carbohydrate metabolism, but I'm very curious about what the protein metabolism is all about when it comes to the Krebs cycle. I'll be sincere, guys, I haven't heard about the importance of Krebs cycle to carbohydrate metabolism and nucleic acid metabolism. I am very fascinated, but I'm sure we have more to talk about when we come to the protein metabolism. And here with me are my colleagues, which are actually going to be diving into that aspect of protein metabolism. Please, can the audience get to know you?
My name is Emidio.
Okay, my name is Wisdom.
My name is Janabasi.
Okay, great. Emidio, Wisdom, and Janabasi, welcome to this podcast session.
Thank you.
Let's dive into. We've heard a lot about carbohydrate metabolism. We've heard a lot about nucleic metabolism. Yeah, so I'm curious, and I'm sure the viewers are curious about the importance of Krebs cycle to protein metabolism. Emidio, can you please take us on that?
Okay, so speaking of the importance of Krebs cycle to protein metabolism, we've been hearing protein. So let me just talk on what protein is first of all. So proteins are large complex molecules that are made up of amino acids. Now, amino acids are organic molecules that are composed of carbon, hydrogen, oxygen, nitrogen. Yeah. So, first of all, when we take in protein to our body, like dietary proteins in foodstuff, maybe beans or eggs, something like that, we chew. And then this chewing process is going to break the food that we eat into smaller particles such that those enzymes can then act upon them. So from the mouth, those contents get into the stomach. Now when it gets into the stomach, it is met with an acidic environment. This is due to the presence of hydrochloric acid. Now this acidic environment is going to denature the protein. It's going to expose the peptide bonds, it's going to expose it. So the presence of this hydrochloric acid is going to activate an enzyme, a stomach enzyme, pepsin. So pepsin then works on these proteins, these denatured proteins, and then breaks them down to individual amino acids and then smaller peptides. Now these stomach contents, we call them chyme. So this stomach content, or let me say chyme, they get out of the stomach into the small intestine. Now getting to the small intestine, we have the pancreatic enzymes. Those are trypsin and chymotrypsin, carboxypeptidase. So they act on these proteins, further breaking them down into much more smaller particles and then still some individual amino acids. Now, after they are being broken down by these pancreatic enzymes, now the enzymes of the intestinal lining, they're called peptidases, they're going to further break down or like hydrolyze every other peptide into individual amino acid. Now that we've gotten to the amino acid, this is where we can then talk on where Krebs cycle comes in. Before amino acid gets into the Krebs cycle, it has to be deaminated. Now, deamination is a process whereby the amino group in the amino acid is taken out, leaving just the carbon skeleton. Now, this happens in the liver. When the amino group is taken out as ammonia is really very toxic to the body. So, the liver is going to quickly work on converting ammonia to urea. That happens in the urea cycle. Now, this is where Krebs cycle plays a part in this deamination process. When ammonia is converted to urea, Krebs cycle produces ATP that fails this process. The beginning materials for the urea cycle, we have carbon dioxide, we have the ammonia itself, and we have ATP. Now, this ATP is provided by the Krebs cycle. So, during this process, Another substance comes in aspartate. Now, aspartate too is produced by one of the intermediates of the Krebs cycle. So first of all, we can say Krebs cycle provides energy and then certain intermediates needed to support the urea cycle. It's going to aid in the safe removal of ammonia from the body.
Yeah.
Then another importance is, looking at the Krebs cycle, oxaloacetate is one of the intermediates. It's gotten after malate. If you look at the cycle, after malate, we have oxaloacetate. Now, oxaloacetate stands as a direct link between amino acid catabolism and glucose synthesis. That is going to be in the process known as gluconeogenesis. Now, that's the process whereby the body produces glucose from non-carbohydrate sources. Maybe the person is fasting or starving, and so there's low carbohydrate intake. So the body needs glucose, and then the human being is not bringing in the glucose. So the body is going to have to find another means to produce this glucose so that we can get energy. So oxaloacetate can be converted from pyruvate. So we have pyruvate, and pyruvate can be converted to oxaloacetate through the action of pyruvate carboxylase. Pyruvate carboxykinase. Carboxylase. Which one was that? Pyruvate was pyruvate carboxylase or carboxykinase? So it's going to become pyruvate carboxylase. Pyruvate is going to be converted to oxaloacetate. Now, from that oxaloacetate, it's going to be converted to phosphoenol pyruvate by the action of phosphoenol pyruvate carboxykinase. So we see that this irreversible process from phosphoenol pyruvate to pyruvate is broken down because oxaloacetate comes in and acts as a bridge to convert pyruvate back to PEP, and then we move forward to produce glucose. So we can say that Krebs cycle produces a very important intermediate that helps in gluconeogenesis.
Okay, great. So one of the things I've gotten here from all she said is that Krebs cycle helps us to fast. So for us that like fasting for 7 days, for 21 days, and 40 days, it will never be impossible without Krebs cycle. If there's no Krebs cycle, we can't fast, because how then does the body provide glucose? How then does the body provide glucose, which is used whenever we are not fasting? Yeah. And when Ash was saying, and she was talking, after she said the very first things that she said, I thought she was done. Then she said, this is where the Krebs cycle now comes in. I was like, wow. So I'm now very curious to hear what Peculiar has to say to this importance of Krebs cycle to protein metabolism.
Okay, all right. Thank you, Medium, for laying that foundation. The Krebs cycle is like a hub. We know that it's a hub. Energy is produced there, but it also helps in doing other things in the body. Like the Krebs cycle helps in the biosynthesis of non-essential amino acids. What are non-essential amino acids? These are amino acids that the body does not necessarily need to get from food. Okay. Like it can be produced in the body. So the Krebs cycle helps in this biosynthesis. How does it do? Like it synthesizes it from another amino acid. Like, let me give an example. Alanine. Alanine goes through the process of transamination reaction with a Krebs cycle intermediate alpha ketoglutarate. What's transamination reaction? It's the transfer of an amino group from one amino acid to a Krebs cycle intermediate to give rise to another amino acid and a keto acid. Like this process is amino acid to react. Like no. Alanine reacts with alpha ketoglutarate with the help of an enzyme, alanine aminotransferase. It now helps to transfer the amino group from alanine to alpha kg, and you have glutamate. The carbon skeleton remaining from the alanine is the pyruvate. That's a keto acid. Then subsequently you can still have deamination. The body cannot store amino acid like fat and carbohydrates, so it needs to deaminate some amino acids. Cannot store it for future use, so it needs to be deaminated. You can deaminate it through certain biochemical processes to have to give rise to ammonia. Yes, that's where we have the ammonia and you can still have, when it's deaminated from maybe one amino acid, we have another Krebs cycle intermediate, that's the alpha kg. Then this ammonia can still, ammonia is toxic now. Ammonia is a very toxic compound. The body cannot keep it, so it needs to be detoxified. So you need to it enters the urea cycle. And in the urea cycle, we have another Krebs cycle intermediate as a byproduct, that's fumarates. These fumarates can now be shuttled back to Krebs cycle to help in energy production. So the entire system is like intertwined That's
yeah. It's linked.
yes, it's linked. So the Krebs cycle helps in amino acid metabolism through synthesis of non-essential amino acids, and it also helps in energy production in the body. So you could agree with me that the entire process is just linked.
Yes, it is.
Yeah, so viewers, you have heard it, the entire process is just linked. Just like I said before, Krebs cycle is a hub of different metabolic pathway. So, Diana, do you have more things to tell us about the Krebs cycle and its help to protein metabolism?
Yeah, I'll be talking about enzymes regulation. Concerning enzymes regulation, the Krebs cycle helps in regulating the enzymes in the pathway through a series of important or delegate enzymes in the production of energy. And one of these enzymes that acts is the citrate synthase. This citrate synthase is an enzyme that catalyzes the reaction, the carboxylic reaction between acetyl-CoA and oxaloacetate.
Yes, oxaloacetate.
Oxaloacetate to give citrate, yeah. And this reaction is allosterically inhibited by ATP to yield high energy compounds. We also have the isocitrate dehydrogenase. This isocitrate dehydrogenase undergoes a oxidative decarboxylation in order to yield alpha ketoglutarate. Yeah. And this alpha ketoglutarate is being inhibited by ADP, that is adenosine diphosphate, in order to provide energy that is needed for the cells. Then last we have the alpha ketoglutarate dehydrogenase complex. Yeah. This alpha ketoglutarate dehydrogenase undergoes an oxidative decarboxylation also, so give isocitrate, and this isocitrate is being inhibited by ATP, succinyl-CoA, and calcium ions. In general, what I'm trying to talk about is that the Krebs cycle undergoes a kind of a regulation by the enzymes in order to promote this amino acid derived carbon skeleton in order to produce energy.
Okay, great. I like the point of the analysis because everything I've heard here today talked about intermediate, oxaloacetate intermediate. But General Abasi came forward to portray the point about the enzymes. The enzymes actually involved in the Krebs cycle, the enzyme. So I've heard a lot today, and I'm sure our viewers have heard a lot. To be sincere, of everything I've heard today, I will not be able to, sit down and learn and portray the points under one day. And I'm actually wondering how many days it will take our viewers to actually, sit down and assimilate these points. All this cannot be done under one day. So
Yes.
I'm going to let our viewers go home right now and assimilate this point that have been said. Till meet again next time. Stay tuned.